Hello. So I wanted to keep talking a bit about all the different tools that we use in marine biology these days. And I actually used a whole lot of them to do my own PhD research. So I wanted to talk a little bit about that. So again, we talked about all the different tools that we used way back when and now. So when I introduced myself, I mentioned that I did my undergraduate work at UCLA, and then I did my PhD research at UC Santa Cruz, studying coral reef ecology and education. And I focused on the recovery of pearl oysters on the coral reefs of Midway Atoll. Well, here's actually a few slides from my PhD dissertation back in 2013. I've worked mostly on education since then, so I haven't done a whole lot of marine biology research since then. So one reason I was super excited to get back to talking about marine biology with all of you. So I did part of my research on marine biology, and I did part of it on education, and specifically integrating math into biology education. I'm not going to talk about that today. This picture in the background is Midway Atoll, where I did my research, and it is famous from World War II. There was a battle there, and it's also an emergency runway for planes flying over the Pacific, as you can see here. So if you've ever been to Hawaii, you've probably been to the main Hawaiian Islands, like over here. That's the main part people know about. But the Hawaiian Islands actually stretch a lot more to the northwest. And much of the northwestern Hawaiian Islands, as you can see enclosed in blue there, are part of the Papahanaumokuakea Marine National Monument. It was established by President George W. Bush and then expanded by President Barack Obama. And it is a totally amazing place where very few people get the chance to go. And I was really lucky to be able to do my PhD research there. I worked at Midway Atoll, way up here, which is a really totally amazing place. It has a great history, like I mentioned, about World War II, and a Navy base for years after that. It also has a lot of really amazing creatures. It has the largest colony of the world of lace-end albatrosses, as you can see here. They are everywhere if you go at the right time of the year. There's also a lot of really cool marine organisms. This is the Hawaiian monk seal, a very endangered species, but we have quite a few of them at Midway, so we saw them almost every day. So I focused on bivalves. So I just wanted to point out bivalves are really important for a variety of reasons. I won't go into detail, but I wanted to make sure you saw that. So I focused on the black-lipped pearl oyster, which had been fished in the northwestern Hawaiian Islands for pearl buttons, as you can see here back in the 1920s, before we made all of our buttons out of plastic, before we really had plastic. And they were harvested so much and so heavily that they almost disappeared. And so the state of Hawaii actually banned, might actually be before it was a state, but anyway, they banned the harvest in 1929, and that ban continues today. However, in the northwestern Hawaiian Islands, people have done surveys, and the species really hasn't recovered since then. And scientists didn't really know why or how to help them recover. So that's what I wanted to do for my PhD research. The managers of the Papahanaumokuakea Marine National Monument, which include the U.S. Fish and Wildlife Service, the U.S. National Oceanic and Atmospheric Administration, and the state of Hawaii, all were really interested in how to restore this species. And it turns out it might not actually be that hard to do, because the same species is farmed in French Polynesia to make black pearls. So there's actually some pretty good strategies out there of how to grow great numbers of this particular species. So I was trying to learn from that and use those same methods. So here's what I did. Pearl oysters, like many marine species, just put their sperm and eggs out into the water that then fertilize and become larvae, which are just kind of little creatures swimming around in the ocean. And eventually, when they get big enough, they'll settle out onto the bottom somewhere. And they're usually looking for some kind of hard place, or hard, dark place. And so in French Polynesia, at the pearl oyster farms, they use the shea cloth that you might see in the hardware stores, often used for gardening. It's like a plastic cloth with holes in it. And so I put out a whole bunch of this folded shade cloth that I called a spat collector. It was floating in the above the reef at a whole bunch of different places. I had about 10 of them at each of these places all around Midway, to try to figure out where the best place would be to collect these invertebrates and to document how many of these invertebrates or these larvae were settling out every time. So this settling out process is called recruitment. And I also thought that probably the biggest risk of mortality to these pearl oysters was as juveniles when they were eaten by predators like fish or octopus or sea stars or crabs. And so I did some experiments where I put some of my juveniles out on these tiles, and I had cages with different mesh sizes around them. So I glued them onto these tiles. Like you can see, this is the baby pearl oyster. And these are some of the potential predators. And I also wanted to know how many adult pearl oysters were out there. So I categorized the atoll into some different habitats, and we'll talk more about these different habitats later, but it comes in handy right now. So if we do a transect across the reef, like you can see here, if we drew a line across the reef and looked at it this way, the highest point where the reef actually almost touches the surface is called the reef crest. It's where the waves are usually breaking from offshore. So here is inside the atoll, and then here is outside the atoll. And outside the atoll, we have the forereef. There's waves crashing there, so we tend to see a lot of like hills and valleys that are formed by the waves. And it's super cool, really amazing place to go diving if you get a chance to dive in forereefs. I highly recommend it. And then inside the reef crest, we have what's called the back reef, which often has the highest diversity of corals. And then farther inside, kind of in the middle, we have what's called the lagoon, which has these patch reefs sticking out. And this actually was where we found the most pearl oysters. And it has this one particular kind of finger coral at Midway. And then surrounding the patch reef, we have just sand, which we didn't expect to find pearl oysters on because they like hard substrate, but we still had to do surveys there. So I used a satellite image of Midway and GIS, Geographic Information Systems, to categorize every little pixel on this image as one of those 4 habitats. So here's what I did. And I only went out to about 60 feet because I wasn't able to go diving deeper than that. So that's why some of the areas are white here, and then this is clouds in the original image. And then of course this white is land, and this blue is inside the artificial harbor where I didn't do surveys. So the purple is the forereef, and then the green is the back reef, and then the red is different patch reefs, and then the yellow is the sand. And I also categorized it into these different sectors as well as the habitats. And I conducted 108 surveys spread out in these different sectors and in these different habitats. And the way I did these surveys was when I was using GIS, I would randomly select, I think it was about 8 points for each habitat in each sector. And then I would put that particular location, latitude and longitude, into my GPS. And we would go out on the boat to that point. We would drop anchor right at that point. And this is the anchor line connecting the boat to the anchor. And then we would go down to that point at the anchor, and we would lay out a transect line, which is like a big measuring tape, for 100 meters, which is about 300 feet. And then we would look for 25 meters, which is about 75 feet. And then we would look for 2 meters on either side of that transect line for any pearl oysters and other bivalves. So that's clams, oysters, other kinds of mollusks that have 2 shells. So that's for a total of 100 square meters. And then we would record the size and number of all of the bivalves that we could find. So these are the main bivalves that we found. My blackfoot pearl oyster, but also this cliff oyster and pen shell, and other oyster. And so this is what my results usually looked like. I got an average density number per 100 square meters per survey for each habitat and each sector. And like I thought, I didn't find too many bivalves in the sand, so that's not even on here. Didn't find any, I don't think. And so for this particular species, the pen shell, Streptopinna, you can see that I found the most of them in the purple area, which is the forereef, so outside the reef. There are different patterns for different species, but I just wanted to show you the results for one. And I was able to use that information to calculate a total abundance for each species. So we had a lot more spondylus than the other species. And you'll notice my pearl oysters, Pinctada isn't even on here. We found so few of them that it wasn't even worth putting on the graph. So you can see I used a lot of math to calculate this, as well as the computer science and GIS to make this image, as well as boating and scuba diving. We also used math to calculate how quickly these bivalves could filter all the water in the lagoon. So we used the same satellite image to get a depth of the different areas in the lagoon. So darker is deeper, and calculate a volume of the whole lagoon. And then I could use the filtration rate from the literature and the number of individuals that I calculated to figure out how quickly bivalves could filter the whole lagoon. And it looked like it was more than a year. So the water was probably moving in and out of the atoll faster than the bivalves could filter it. So it looks like the bivalves probably were not filtering the plankton. We're not limiting how much plankton there were. So with all those surveys that we did, we actually found in the surveys and in other work that I did in the 7 years that I worked at Midway, I think, we only found 16 adult pearl oysters. They're pretty big. They're like a dinner plate, like you can see here. And so most of them were on these patch reefs. We did find a couple inside the harbor and one out on the forereef. But we found hundreds and hundreds of juvenile pearl oysters on my spot collectors here, as well as in other places. So there are 2 pearl oysters that are reported in Hawaii. There's the one I was looking for, Pinctada margatifera, which is really big. Like you can see, it's like the size of a ruler or even bigger. And that's what it looks like. It's a juvenile. And then there's also oh, sorry. These are the adult, these are the Pinctada margatifera. And then these are the second species, Pinctada radiata. Forgot. It's much, much, much smaller, even as an adult. So it's really hard to identify juveniles as an adult, as a juvenile. So my pearl oyster expert said, you better check that all those juveniles you're getting are actually Pinctada margatifera and not Pinctada radiata. So then I had to learn how to do genetics, which I had not studied. I considered myself an ecologist and I didn't know anything about genetics. But in order to figure out which species it was, I had to do genetics on my samples to figure out where on the phylogeny or family tree of pearl oysters they fit. So here's the Pinctada margatifera, which I thought I was looking for. Here's the Pinctada radiata. That's the other small species found in Hawaii. And then these other species are found in other places throughout the Pacific. So I used some genetic analysis methods. I made lots and lots of copies of the particular sequence that I was interested in using PCR. And then we used sequencing to figure out what the actual sequence of T's and C's and A's and G's were at this particular marker. It's ITS1, in case you're interested. I believe it's on the ribosomal DNA. So I use this old method. There's a lot fancier methods now, but I didn't use that. In fact, I'm not even 100% sure. I only did the PCR part. I paid someone else to do the rest of it. So here's what the phylogeny looked like using data that was in GenBank, except for my samples. And I used a computer program and programming to figure out where my samples fit. So here's my samples. I was able to get 17 samples that I got a good sequence from. Here's the Pinctada margatifera, the blacklet pearl oyster that I was looking for. Here's the other species found in Hawaii. And you notice my species actually aren't all that close related to either of them. which is a little bit weird. They're actually most closely related to Pinctada maculata, which had not previously been reported in Hawaii. So it turns out all those juveniles I was getting are actually not even the species I was looking for. So you can't really restore a species with a totally different species. So that kind of ended up making me throw away much of my PhD research. My PhD ended up being the surveys of all the different bivalves, this genetic research, and then also a chapter on education. Science often does not go the way you expected or the way you planned. Let that be a lesson for your future. Most things don't actually, but especially science, and especially working with wildlife. It can take forever and not work and be completely frustrating, but also really amazing when things do work out, or when you are able to accomplish something despite all that. So my juveniles that I was finding are actually Pinctada maculata, not previously reported in Hawaii. So what do we know about this? So they're actually kind of considered, they have the mother of pearl inside the shell is kind of golden. And so they actually make these kind of golden pearls in the Cook Islands, these really, really tiny gold pearls called PB pearls. And they are found in different parts of Asia and the South Pacific. But there was one report in Hawaii, which surprised me. I don't think that was my report, but I hadn't heard that before. And I have no idea how it was reported in the United States, according to this database. But so how could this Pinctada maculata get to Midway when they've never been seen before? Well, it's possible that they were there all along and just nobody ever noticed it because they're pretty little. And there hasn't been a lot of work done in the northwestern Hawaiian Islands. It's possible that they were misidentified as Pinctada radiata because they're both small pearl oysters. And it's possible that it's actually an introduced species that just hadn't been noticed yet, which is called a cryptic invasion. So there's different reasons to support these different things. So there were 3 main bivalve species at Midway. Most of them are more common on the Reef. They're probably not limiting how much plankton there is at Midway. The black-lipped pearl oyster as adults are extremely rare. Their recruitment must be pretty small because all of the juveniles that I was able to test were actually this other species, Pinctada maculata. So let that be a lesson. If you're trying to do science or conservation, make sure that you carefully identify the species you're working with. So that's it. I hope you enjoyed learning about my PhD research.